Hello everyone, my name is Owen Visser and I am a student at the University of Florida. I work with Dr. Sambath Data in the Data Lab group, and today I am giving a short presentation on the measures for the evaluation of clustering methods on single-cell data. As we all know, single-cell data has been growing in availability, and the number of research papers that have been published recently has gone up quite a lot over the last couple of years. And one of the major pieces of research going on in this field is the identification of cell clusters. Validation of these measures is challenging, often overlooked, and it usually leads to less critical choices in clustering methods, which is used for cell identification. And so the idea then is we want to validate these measures, or these methods, using validation measures and techniques. Usually this is done through post-analysis with external measures, but we don't really know the true biological groups all the time, so using these external validation techniques is not always a possibility. Furthermore, post-analysis methods are subjective to dataset, and so we need to have some sort of method to validate these internal and external these methods internally and externally for each dataset in a quick fashion. Back in the field of microarray data, Dr. Data had a similar issue that he solved with his colleague Dr. Brock and a student Picard. They solved this by creating a package called CLValid, which used a group of internal and external and stability measures to evaluate clustering methods. For single cell, though, we can't directly adapt the equations because there are complications in the data set via extreme amounts of correlation and the way that the samples themselves, which are the cells, are clustered. Back in microarray, the genes were clustered and the samples were removed to instigate stability, but here we are going to be removing the samples, which is the cells themselves, to instigate stability, which we'll show you how we do that in a little bit. The method is called leave one out, which is very close to jackknife, stems from the same basis. All right. So the objective then is to demonstrate the utility of the internally adapted measures from both CLValid and the ones that are going to be subjected onto single cell data. And we want to also make sure that we are excluding the value of external validation. Okay, so on the screen here we see orange circles and blue circles. These are going to be the true groupings of the cells, so cell groups. Whenever we see a dotted circle, this is going to be a statistical clustering, and whenever we see a line, this is just an indication that we're in 2 dimensions and it's a sort of distance. Okay, so the first type of clustering measure is the external measures. External measures are quite simple. Basically what we're doing is we're looking at the comparison of the true cluster, the statistical cluster, and its correct assignment, right? Whether or not the true cluster and the statistical cluster match. So we have measures like purity, which is simply just the number of correct true cluster assignments that we've done, and then the rand index, which is just a probability of assigning both the true to the positive and the true to the negative cluster assignments. Next type is internal measures. Internal measures usually come in 3 forms. Compactness is the first form, which measures variations of samples from a central location, as we see here. Connectedness, which measures the between distance of individual samples in a cluster. This could be one cluster or one cell to the next cell within a single cluster. It could be a shorter distance between cells. It could be a longer distance between cells. It's just the distance between individual cells within a cluster. And then separation, which is how far apart different clusters of cells are from one another. This could be the closest cell, as we see here, or the furthest cell, as we may see later. Of these 3 major groupings, compactedness, connectedness, and separation, comes our measures. There are very simple measures and there are very complex measures. Most measures are combinations of the 3 above, so something like the Dunn index, which is our example here, is the minimum distance between clusters ratioed to the maximum distance within clusters. This is a comparison of connectedness and separation. Notice that we don't have any compactness here, but some other measures of combinations do have compactness, such as silhouette width, connectivity, and group proportion. One more complex type of internal clustering is going to be what's called partition compliance. This is what happens when we remove the cell from the clusters and then regroup and see what has changed. So as we see here, if I go back slightly, we can see that when we remove this cell that's in red, we then recluster and we find that this one blue is in the wrong spot statistically. This is done after dimension reduction. If we instead do it before dimension reduction, we actually end up shifting all of the information from each of the remaining dimension reduced sample points. Then when we recluster, even though that is removed, we actually see we have the correct statistical clustering. So this is an idea of rather partition compliance, but stability, because we are removing information, not just the sample. We're removing the sample, and then along with it, all of the information that would be spread to everything else. This is the basis of all the measures that Dada used for his package CL valid, the stability measures that is. And with this adaptation of partition compliance before dimension reduction, we can actually adapt these measures to work in the same fashion on single cell data for groupings of cells. So that's the general idea. We want to use this kind of method so that we don't have to remove any genes to initiate stability, because genes are the most important thing when it comes to these. I mean, we want groups of cells, but mainly we want to understand what genes are driving these cell groupings, because that's ultimately what we want to change, or what we want to identify. Not just the cell types. Knowing the cell types would be great, but knowing the genes that make up the cell types is more important. If we start removing genes to change the information, then we remove all of the beauty of feature selection. So we want to retain that. Obviously, this is going to help with some computational efficiency as well, because with being able to keep feature selection and dimension reduction, we're actually going to be speeding up the process of identifying these clusters as well, which is great. And so the stability measures will retain their function. So I want to go over a few of the stability measures now, and we'll see what they do. So before we do that, actually we need to go through a little bit of notation. So as we said, the colors are the true cells. We have n true groups denoted by T, so we have T1, T2 here. Tj is going to be the group missing cell J. As we see, this more transparent circle, the orange, is the missing cell J. And then Ti will just indicate a group that has cell I. So cell I is here, Ti is the true group containing cell I. We also have this kind of notation for statistical clustering. So there are L many cells, but here we have K many clusters. We have C1, C2. So cluster 1, cluster 2. C minus J is going to indicate the cluster based on data that was clustered and then cell J was removed afterward. See here, the transparent one is cell J is removed. And then Cj, not C minus J, Cj now, is based on the removal of J, Cj is removed, and then we proceed to statistically cluster, and these would be Cj. So we have C11, C21, right? Cell 1 was removed. And then once again, Ci is the cluster that contains sample I. So we have cell 2 here, C2 would be, or C in parentheses 2 would be cluster 2. All right. So average distance is the first measure we're going to look at. And what average distance does is it measures the compactness of clusters by computing the average distance between genes expressed profiles over the before and after clustering. I'm saying before and after because we have before removal of J, after the removal of J. So here we're going to pretend this is the after removal of J. And so what we're going to do now is we're just going to change J from before to after dimension reduction, and we're going to see the new locations. So the opaque ones are going to be the after, and the transparent ones are going to be the before. And we're here comparing the distance between the cells in the new and before cell I grouping. Next, we have adapted or average proportion of non-overlap, which is very similar to the distance. Instead of measuring the distance between the cells after we remove cell J. Instead, we're going to be looking at the intersection. So here we see that the intersection is very small, so this is not going to be a good measure. Moving to the last measure, we have biological stability index, which is going to measure the before and after states of cells across true groups or within true groups. So here we have cell X and cell Y are within the same true group. So we're going to be looking at how they move. So cell X is over here, cell Y is over here after they move. And so we're going to compare the before cell Y and cell X in their respective cases. So here we're looking at the after location of cell X and the before location of cell Y, and we're going to be looking at the intersection there. And then here we're going to be looking at the intersection of the before cell X and the after cell Y, and look at the intersection there. Alright, so these as an example of 3 of the measures. Here we're going over the gold standard data sets that were used to validate these, or to show the usefulness of these measures. So we decided to go with 5 gold standard data sets, and what gold standard data set means, it means these are data sets that are widely accepted and considered extremely accurate, and these data sets in fact were looked at visually through a microscope to make sure that the cell groupings were correct. These are examples of embryo and cortex cells. The embryo cells, more specifically, are easily identified because you can look at the individual cells splitting. So you can look at the 1 cell, you can look, or the zygote, and you can look at the 2 cell state, then the 4 cell state, then the 8 cell state, and so on. And so we have these groups. Notice these are quite small data sets, but that is just due to efficiency to get a notion of whether or not this is effective. For each data set, we decided to do some very basic pre-processing. We didn't want to instate any feature selection to any single group by itself or to over all groups, because each of them have their own special little feature selection they do and dimension reduction, so we didn't want to go through too much of that. So we decided to just filter out the non-counted genes, normalize everything, and scale it to log scale. For base methods of dimension reduction, we decided to use PCA and t-SNE, and we'll go over that in a second here. For clustering methods, very basic ones, we decided to go with hierarchical agglomerative using the word linkage. We just wanted to go with a very basic agglomerative method, which means we started with each individual cell and grouped them based on words linkage, and then for the desired number of clusters, we cut the tree. For k-means, we used Hartigan's base method from 1979, where we just choose a number of centers, and cells are continuously and iteratively changed centers or changed groups in order to the distance of their center until we reach the desired number of clusters. We also used race ID, which is a method in 2015, which is Hartigan's k-means, but instead of using on PCA, we decided to use it on, this method uses on Pearson's correlation coefficients on the count matrix. CIDR, which is in 2017 by Lin et al., uses k-means, but instead of on either Pearson correlation or PCA or t-SNE, it uses it on dimension kernel density approximation of the variance. And then we also have PCA reduce, which is a method in 2016, which uses an agglomerative clustering method on top of k-means. So we use PCA and k-means to get a set of clusters, and then using a hierarchical method, both probabilistic and bootstrapping, they would pair the 2 closest groups together and repeat this until every single number of clusters is done from k all the way down to 2. Alright, because we have such an insane number of methods and measures and cluster sizes for each data set, we end up having a completely ridiculous number of measures, so we need to use some sort of package in order to rank these and identify what is the best method available. So we use Dr. Dada's and Per's package from 2009 called rank-agreg, and what this will do is this will order the standardized measures into a nice table, as we see here. So how it works is we end up ranking each of the cluster sizes for each method, and using this information we can go ahead and apply the overall rank of cluster size to weight the next table, which ranks cluster size or ranks the methods based on each cluster size, giving us an overall best method. This overall best method can be seen performing the best at specific cluster sizes, and this is how we are going to identify what cluster sizes each method can or performs best at. So based on our results, we find that if we use solely the adapted methods, we have that the PHC method performs best at a cluster size which is included in the number of true groups. For conventional only, we find a single method or a single data set worked. For both both adapted and conventional methods, being all of the internal methods, we were able to identify the best method pertaining to the true number of groups for 4 of the data sets, and similarly if we include the external measures as well. What this has showed us is that the adapted measures do perform better than conventional methods, measures for identification of proper clustering methods. However, the more important thing we know is that when we pair both the adapted and conventional methods, being all the internal methods, we find a relatively equal number of best methods at certain cluster sizes when compared to using external measures, which is the goal. In the end, we want to make sure that using only internal measures, we can pick a good method. So in conclusion, we've adapted some validation measures to use for statistical method choice for single cell data. Using rank aggregation, we were able to find which method performs the best and at which cluster size they perform best at. And the code used to do this is quite quick. It's mostly written in C++ and it performs quite fast, especially the measures, and will soon be publicly available in a package. And furthermore, it offers a decisive approach to clustering method choice. As far as the use of Seurat and Monocle, we did not choose to use these, mainly because Louvain and Leiden clustering did not let us choose the number of clusters, but we are right now currently writing code in order to compare all of these methods together with choice, so that is going to be done. As far as simulated data can't calculate or can't capture all the nuisances, or not nuisances, nuances, so nuances of single cell data. So that's why we decided to not use simulated data. And lastly, we are going to be expanding this for even larger data sets. Like as I said before, some of these data sets are quite small, so we want to tackle larger data sets. So instead of using a leave one out, we might end up using some sort of k-fold validation strategy for stability. I just want to say thank you to Dr. Dada and the Dr. Dada Lab group, as well as CAMDA, for allowing me the opportunity to speak. Thank you very much.